So this study was performed in collaboration with Dr. Robert Zavadanov and Dr. Dehan Jakimovsky and their colleagues at the State University of New York, Buffalo Neuroimaging Analysis Center, or BNAC for short. We've had a long-standing collaboration with them that's focused on serum proteomic associations with multiple endpoints of disease progression. This includes clinical, neuroimaging, OCT and MRI, and neuropsychological assessments as well. For this poster, the serum proteomic work was performed by Octave using our custom assay panel. That's a panel we've developed and validated that measures the concentration of 21 proteins. Those proteins were selected based on association with MS endpoints of disease activity as well as disease progression. The statistical analysis for this particular poster was performed by BNAC, and it investigated proteomic associations with exploratory imaging biomarkers, which included ALV, or atrophied lesion volume. This is the volume of lesions that have subsumed into CSF, predicting progression and transition into MS phenotypes, as well as LMCE, or leptomeningeal contrast enhancing lesions, and those reflect greater cortical pathology. So the samples from this study were from the CEG cohort, approximately 200 patients with MS that were followed over 5 years. And we had deeply phenotyped data, extensive data collection and annotation, along with the serum samples. This happens to be one of the most extensive cohorts we've evaluated at Octave. We were excited to find associations of 2 proteins on our panel with atrophied lesion volume. This included GFAP and MOG. These were associated with greater accrual of atrophied lesion volume over the follow-up period. Our overall conclusion from this poster was that biomarkers of glial activation, and both overexpressed and underexpressed in patients with progressive worsening MS, may be specific to the progressive phenotype. Many findings have come from this study. We earlier reported associations with clinical endpoints, EDSS, standard MRI metrics, diffuser tensor imaging, and neuropsychological assessments were also reported at ACTRIMS this year. Our next steps are OCT. We have very extensive OCT data on these patients and potentially looking at 10-year outcomes in this cohort. So briefly, there was 202 patients that were followed over a 5-year period. And for this poster, we looked at the serum proteomic associations with neuropsychological tests that are used to assess cognitive and functional disability in MS patients. So we looked at 9-hole peg test, PASAT, SDMT, and the timed 25-foot walk. We found that the 9-hole peg test, SDMT, and timed 25-foot walk were strongly associated with disease progression as defined by EDSS. Several proteins from our custom assay panel were significantly associated with worsening neuropsych test outcomes: CCL20, neurofilament light, GFAP, CDCP1, and CXCL9. Notably, these proteins represent diverse biological pathways. I mentioned that earlier we had evaluated clinical and standard MRI assessment, so brain volume, gadolinium-enhanced lesions, T2 lesions, as well as exploratory radiographic endpoints, diffusion tensor imaging, and we just discussed atrophied lesion volume and leptomeningeal contrast enhancement. This is a very deeply phenotyped cohort with long-term outcomes, and evaluating a diverse set of endpoints relative to serum proteomic associations can really help drive the field forward in understanding, predicting disease progression, identifying subtypes of patients, and ultimately using these findings to support drug development and clinical monitoring of patients.